Hello, MitOx community. I am Bhupesh and I am an infection biologist from Würzburg, Germany. Today I am going to talk about pathogens and mitochondria. Two of my most favorite pathogens that I am going to talk about today are chlamydia and human herpes virus type 6. If you have questions regarding our work, please feel free to drop me an email at the address mentioned here.
We have been taught to understand pathomechanism of human diseases through linking them to a specific pathogen. But in reality, we live almost a symbiotic life. It is quite possible that multiple pathogens, some of which in fact live within our cell, can interact and influence each other, which in turn can shape the fate of the diseased cell. I'm going to share today our experiences working with chlamydia, which is an intracellular obligate bacteria, and several different types of human herpes viruses, including human herpes virus type 6 or HHV6. I will show you how latent herpes viruses like HHV6 reactivates after chlamydia infection and how mitochondrial architecture play a crucial role in the life cycle of both these distinct pathogens.
We all know that mitochondria plays both positive and negative roles in the life cycle of pathogens. Those pathogens who need a strong metabolic input from the host cell for their life cycle tend to strengthen mitochondria, whereas those who prefer to escape from innate immune response originating from mitochondria tend to negatively influence mitochondria. And all this happens through alterations in mitochondrial architecture, through fission and fusion of mitochondria. Here in a video, I am bringing your attention to dynamic nature of mitochondria in our cells. This dynamic nature of the mitochondria is also responsible for cell danger response that in turn regulates pathogenic growth and health of the cell. As you all know, mitochondrial dynamics is regulated by multiple pathways. However, one of the most important regulator of mitochondrial architecture is a dynamine related protein 1 or DRP1. DRP1 is transcriptionally regulated by the transcription factor P53, which in turn is regulated by human microRNA family MIR30. When MIR30 is upregulated, it negatively regulates proteins like P53, caspase-1, and caspase-3, which in turn brings down the protein levels of DRP1. This situation favors mitochondrial fusion. However, when MIR30 goes down, it upregulates proteins like P53, caspase-1, caspase-3. Because of the upregulation of the P53, DRP1 is upregulated, and this situation favors mitochondrial fission. I call DRP1 as the Lord of the Fission rings. DRP1 is a GTPase which forms ring-like structures around mitochondria and clips them into smaller fragments. We can in fact measure the number and structure of these DRP1 rings around the mitochondria and can quantify the state of the mitochondrial fission in a given cell. Now let's see how chlamydia manipulates mitochondria and why it does so. Chlamydia infection is one of the most frequent bacterial infection in human being. Chlamydia trachomatis infection is one of the most frequent sexually transmitted disease. Chlamydia has a biphagic life cycle in the form of elementary bodies or EBs and reticulate bodies or RBs. Here in this structured illumination microscopy image, you can see the chlamydia EBs and RBs inside a human cell in culture. Chlamydia is a gram-negative obligate intracellular pathogen and it is a very strong energy parasite. It requires high amounts of ATP from the host cell to survive. It prevents initiation of apoptosis and it induces reactive oxygen species production, modulates expression pattern of several host microRNAs, and pro-apoptotic factor including p53. Chlamydia infection protects the host mitochondrial architecture by allowing mitochondrial fusion. Here in HeLa cells, you can see a different mitochondrial morphology in cells having chlamydial infection. You can see those pink elementary bodies in infected cells. The situation is even better in human ovarian fimbriae cells of primary origin, which are natural sites of Chlamydia trachomatis infection. Here in another image, you can see similar mitochondrial morphology preferring mitochondrial fusion in human umbilical vein endothelial cells. When we looked into the microRNA transcription profile using next generation sequencing in chlamydia infected cells, we saw major changes in human microRNA. On the left panel, you can see a heat map showing upregulation of several human microRNAs after chlamydia infection. One of the prominent upregulated human microRNA was MIR30C. This upregulation was significantly high after 24 hours of chlamydia infection. On the right hand side, the same was validated by northern blotting in 3 different cell types that confirmed a sharp increase in mature MIR30C levels post 24-hour chlamydia infection. Then we asked the question if silencing MIR30C would interfere with the growth of chlamydia. For this, we first created an inducible microRNA 30C sponge expressing cell line where a microRNA sponge can be expressed under TET inducible promoter. When we added anhydrous tetracycline to these cells, we silenced microRNA 30C that resulted in increased p53 expression and fragmented mitochondria. As you can see, chlamydial development was severely affected under these conditions, which was evaluated by quantifying chlamydial OMP-A protein and counting chlamydial inclusion bodies. Strong downregulation of chlamydial growth was observed under microRNA 30C silencing conditions. To validate that DRP1 and mitochondrial fusion is key to chlamydial growth, we evaluated DRP1 protein expression during chlamydia infection. As you can see, DRP1 protein levels were decreased upon chlamydial infection. When we overexpressed DRP1 using a pcDNA-based vector, chlamydial growth was inhibited. On the other hand, when DRP1 expression was silenced using siRNAs, chlamydial growth was enhanced. In fact, chlamydial infection could not increase the DRP1 expression as well as its aggregation on mitochondria even under hydrogen peroxide and cobalt chloride stress. As you know, hydrogen peroxide and cobalt chloride are strong inducers of mitochondrial DRP1 aggregation and hence mitochondrial fission. We counted the DRP1 mitochondrial aggregation or rings to quantify this effect. Till this point, it sounds like a very usual microRNA based study. Let's give it a twist. What if these chlamydia infected cells carry latent HHV6? I am a virologist and naturally I see viruses everywhere. But that's not the reason why I love HHV6. These are the only family of viruses that can be genetically inherited. In fact, 0.2 to 1% of human population inherit 1 to 2 copies of fully intact HHV6 genome from their parents. This number can be staggering, up to 2.6% in Scotland. This is because HHV6 carries a human telomere repeat at both ends of the viral genome and hence prefers to get integrated into subtelomeric regions of human genome. Besides this, almost 90 to 95% of the human population gets HHV6 infection between the age of 1 to 3 years. Under these conditions, the virus remains latent at random tissues in our body, including brain tissues. Most of the times, these latent viral genomes are functionally inactive. However, Under various physiological conditions, including immune suppression, these viral genomes can come out of the chromosome and can become active again. During our studies with chlamydia, we observed a very interesting phenomenon. We saw that chlamydia infection induces telomere shortening. In human cells, the telomere shortening is transient and gets repaired within 8 to 10 hours of shortening. Both Chlamydia trachomatis and Chlamydia pneumoniae cause this effect. By the way, Chlamydia pneumoniae causes a lung infection. In murine cells, this repair seems to be defective. This can also be quantified by FISH. As we know, telomere shortening creates telomeric D loops. If there is a latent viral genome within telomere, it can also come out in a circular form during this process. In fact, by metabolic labeling and 2D DNA electrophoresis, we could show changes in telomere structure upon expression of one of the chlamydial protein called Nu. You can see that Nu protein expression in HeLa cells suppresses single-stranded and supercoiled telomeric DNA and increases the formation of open circle telomeric DNA. So, what happens when HHP6 reactivates? We could show this reactivation of HHP6 prefers fragmented mitochondria. In these images, you can see the virus reactivating cells in red. These cells carry a RFP expression cassette within viral genome. Because of the viral latency in telomeric regions, which is a silence region of the human genome, we do not see RFP expression in the cells when virus is latent. Upon virus reactivation, cells start expressing RFP. This is like a reporter cell. You can see clear differences in mitochondrial structure and architecture in the cells which have reactivated HHP6. Here I show you a closer look on the mitochondrial structure. Latent HHP6 carrying cells show a more filamentous mitochondrial structure, whereas upon virus reactivation, a more punctuated mitochondria can be observed in these cells. Here are some more images from structured illumination microscopy showing a completely different form of mitochondria in virus reactivated cells. These cells were reactivated for HHP6 using a histone deacetylase inhibitor called trichostatin A. We carried out direct infection of HHP6A in Hubic cells and virus reactivation in U2S cells. Under both these conditions, we observed upregulation of p53 both in direct infection and in virus reactivation. Similarly, DRP1 was also upregulated under both direct viral infection and virus reactivation. In U2S cells, virus reactivation was carried out using trichostatin A, which is indicated here as T. SC stands for solvent control. We could also quantify mitochondrial fission rings in these cells using structured illumination microscopy, which showed increased number of fission rings on mitochondria. We could also show that DRP1 is actually helpful for viral survival. If we use siRNA against DRP1 and suppress the DRP1 levels, virus reactivation remains unsuccessful. We used our MIR30C spawn cells that I mentioned before, which resulted in DRP1 upregulation. Northern blotting results showed that these sponges decreased MIR30C levels in the cells. You can compare this between the second and the third lane. This MIR30C decrease was not transcriptional. It was a processing defect of microRNA 30C as pre-microRNA 30C accumulated in the cells while mature microRNA went down. 30C sponge carrying cells increased the negative effect of microRNA 30C and allowed better viral survival, which was measured by analyzing one of the viral small non-coding RNA U14. Like I showed you before, HHP6 reactivation induced microRNA 30C processing defects. Going ahead, we found that this might be because of a viral microRNA called microRNA U14. To prove this, we created an inducible cell line where microRNA U14 was expressed in the absence of any other viral components. Upon doxycycline treatment, these cells showed similar microRNA 30C processing defects. Pre-microRNA 30C was accumulated in these cells upon expression of viral microRNA U14. For a better understanding, we have shown here pre-microRNA to mature microRNA ratio, which changes under conditions of microRNA processing defects. We then created a cell line carrying a latent but mutant HHV6 which doesn't express MAU14. As you can see here on the left panel, wild-type HHV6 reactivation induces a strong mitochondrial fragmentation. In these images, virus reactivating cells are GFP positive. Mitochondrial fragmentation is indicated with green arrowhead. On the right panel, the mutant virus reactivation has no effect on mitochondrial fragmentation. Hopefully, I managed to raise a very unique question here: to fragment or to fuse? This characteristic feature of mitochondria is crucial to the survival of pathogens. I showed you here chlamydia infection induces fused mitochondria through microRNA 30C DRP1 regulation. However, HHP6 induces fragmented mitochondria using the same microRNA 30C DRP1 axis. When they both meet each other in a single cell, it seems that virus wins while chlamydia enters into a stage of persistency. Finally, I would like to take this opportunity to acknowledge members of my lab and my collaborators. Nitish and Subhagata did their PhD with me and contributed to the success of my lab. Subhagata was very talented and helped me in creating several valuable tools for mitochondrial study. I am also thankful for the excellent core facility at the University of Würzburg, without which we could not have done this work. I am also thankful to our funders for their financial support and also for your attention